Most banded iron formations are produced when dissolved iron reacts with oxygen in the water and drops out of solution as rust. This process usually produces large banded iron formations, much larger than those seen in the Mesoarchean. So, where did the banded iron formations from this era come from? One hypothesized source is photoferrotrophy, or photosynthesis, which uses iron 2 as the electron donor, producing rust. Photoferrotrophy occurs in some modern habitats and is carried out by green sulfur bacteria, which use an iron oxidase gene to take electrons from ferrous iron, which is then energized in their photosystems. Relatively recent algoma-type banded iron formations, similar to those which occur in the Mesoarchean, have been found in Greece, and the molecular signature of green sulfur bacteria has been identified within them, indicating that these were produced by photoferrotrophs. It should be noted, however, that the iron oxidase gene used by modern photoferrotrophs emerged after the Mesoarchean. So, if photoferrotrophy was active during this time, the ancient and modern versions evolved convergently. Oxygenic phototrophs use two photosystems, PS1 and PS2, while the major groups of anoxygenic phototrophs each only use one. Interestingly, the purple sulfur bacteria have a photosystem which closely resembles PS2, while that of green sulfur bacteria resembles PS1. Oxygenic phototrophs split water in a reaction center within photosystem 2, while purple and green sulfur bacteria have enzymes take electrons off hydrogen sulfide, which is then brought to the photosystems by electron carriers, where it is then energized by photons. The photosystem of purple sulfur bacteria has an iron atom in the reaction center, so one way ancient photoferrotrophy could have come about is with a photon striking this iron and stripping an electron off that went down the electron transport chain. The iron would then be replaced by another iron from the environment, get struck by a photon, and the cycle would repeat. Such a system could ultimately give rise to the photosystem 2 complex. Now, let's take a look at the broader ecology of photosynthesis during the Archean. Without an ozone layer, UV light would have inundated the top 3 meters of the ocean, killing virtually all microorganisms. It is true that there is plenty of light below 3 meters, with the majority of photosynthesis occurring beneath that depth. However, the top 40 to 120 meters of the ocean are well mixed by wave action, and since most single-celled photosynthetic organisms take about a day to divide, virtually all planktonic phototrophs in the top mixing layer of the Archean oceans would have spent enough time in the top 3 meters that they would have gotten a lethal dose of radiation before they could reproduce. It is true that we found microbial mats from the Archean which existed in tidal zones, but being attached to the bottom of the oceans, the dose of UV light that these organisms would be exposed to would have rose to lethal levels over the course of several hours as the sun rose in the sky, as opposed to seconds from being washed around the water column. This would give benthic photosynthetic organisms time to enact protective measures such as going dormant, secreting photoprotective pigments, or burrowing into the sediment. So, if planktonic productivity existed in the early to mid-Archean, it would have existed below the top mixing layer, where it would be limited by the quantity of sunlight penetrating those depths. Planktonic phototrophs were unlikely during the Paleoarchean, since they would have been tied to hydrothermal vents which emitted sulfides. However, given that ferrous iron was a major constituent of the ocean's ions at the time, phototrophs could have begun to colonize the open oceans during the Mesoarchean. The banded iron formations of the Mesoarchean are composed of magnetite and hematite. Hematite has 3 oxygen atoms and 2 iron 3 ions, while magnetite has 4 oxygen atoms, 2 iron 3 ions, and 1 iron 2. In other words, magnetite is more reduced than hematite. When the isotopic composition of carbonate inclusions in magnetite and hematite are compared, those in magnetite are comparatively depleted in carbon-13. This suggests that the magnetite was produced by respiring organisms, which use iron 3 as an electron acceptor as they oxidized organic carbon, which usually has less carbon-13 than atmospheric carbon dioxide. This further suggests that dissimilatory iron reduction, or DIR, had evolved during the Mesoarchean, possibly taking advantage of the iron 3 produced by photoferrotrophs. Regrettably, the evolutionary history of DIR is not well understood, but in terms of electronegativity, iron is between sulfur and oxygen on the redox ladder. Utilizing a more electronegative electron acceptor will produce a greater energy yield for every mole of organic carbon oxidized, but there's also a trade-off because these also induce greater oxidative stresses. As microorganisms evolve mechanisms to cope with these stresses, they can be thought of as climbing the redox ladder. Because iron 3 is poorly soluble, iron reducers have to use chelating agents to get the iron into their cells, or have enzymes on the outer side of their cell membrane to shuttle electrons to the iron. In most modern aquatic and marine systems, respiration takes place in the water column, while fermentation happens in the sediment. However, since hematite produced by photoferrotrophs would have precipitated out and sunk to the bottom, you'd likely have the reverse in the Mesoarchean, with fermentation occurring in the water column, while respiration would occur in the sediment. The production of methane has long been seen as a key determinant of climate on the early Earth. A third variety of methanogenesis is known as methylotropic methanogenesis. Here, methanogens metabolize methyl groups on biomolecules as an energy source. To do this, they use a different modification of the Wood-Youngdahl pathway, feeding an organic carbon into where the methanogenic and acetogenic versions diverge. The methyl group flows down the last part of the methyl branch until it's reduced to methane. Meanwhile, another carbon goes up said branch in reverse until it's oxidized to carbon dioxide. Once the biosphere had become more active in the Paleoarchean, methanogens could use this pathway to metabolize volatile organic molecules released by phototrophs. However, the late Mesoarchean saw the evolution of another pathway, methanotrophy. Methanotrophs oxidized methane for energy while using some of it as a carbon source. Methanotrophs began the oxidation of methane by using the methane monooxygenase enzyme. However, when we go further down the pathway, some of the cofactors used in the final steps of methane oxidation are also the ones used in the initial steps of hydrogenotropic methanogenesis, or the final steps of carbon oxidation in methylotropic methanogenesis. Furthermore, many of the enzymes used to synthesize these cofactors are homologous in methanogens and methanotrophs. This suggests that methanotrophs evolved in the presence of methylotropic methanogens, with lateral gene transfer frequently occurring. The first isotopic evidence of methanotrophy dates to 2.9 billion years ago, with sulfate being used as the electron acceptor. The beginning of methanotrophy may have drawn down methane levels in the atmosphere, cooling the Earth and leading to the Pongolan glaciation. So, how these adaptations have manifested themselves in the broader Mesoarchean biosphere. For one, photoferrotrophs would have had a decisive advantage over photothiotrophs because they would have a literal ocean of iron to use as an electron donor. Productivity would have been exacerbated by the ever greater weathering of phosphorus from the expanding continental margins. With iron being a more potent electron acceptor than sulfate, iron-respiring organisms would have also had an advantage over sulfate reducers, which can explain why sulfur mass-dependent fractionation is less common than in the Paleoarchean, but it doesn't account for the decline in mass-independent fractionation. As photoferrotrophs expanded into the open ocean, they would have been limited by UV light. These organisms would release simple volatile organic molecules into the environment, as many algae do today. However, unlike today, where most of these compounds are oxidized by reacting with oxygen, 3 billion years ago, they would have built up in the atmosphere. Here, they would react photochemically with UV light and produce a layer of Tholen haze. This would in turn provide UV shielding to the surface and explain why sulfur mass-independent fractionation becomes less common. This would also allow planktonic phototrophs to inhabit the top mixing layer of the ocean. Something peculiar is that at all depths, chlorophylls used by oxygenic phototrophs absorb less light than bacterial chlorophylls. But if chlorophylls evolved in organisms suspended in the upper layer of the oceans, they would have had to cope with rapid changes in light intensity as they were moved up and down the water column. Sudden increases in light intensity can cause photosystems to blow out. But since chlorophylls have a peak in the blue range and blue light can penetrate the furthest in water, chlorophylls would experience a lower rate of change in light intensity as they were moved up the water column. So chlorophylls may have evolved during this time period. The photooxidation of iron in the water column and its reduction and replenishment in the sediment would have led to a more self-sustained carbon cycle in the ancient biosphere. In summary, the Archean Earth can be chronologically divided into different eras by which element was the main electron donor in primary productivity, with a hydrogen, sulfur, iron, and then an oxygen age during the Great Oxygenation event. Acetogenesis, followed by methanogenesis, are the oldest extant autotrophic pathways. Dissimilatory sulfur metabolism likely began with methanogens reducing sulfite to sulfide in order to detoxify it. This was later coupled to the central energy metabolism, giving rise to the first form of respiration. Sulfur reducers later evolved to metabolize sulfate, thiosulfate, and elemental sulfur. Photosynthesis likely began as a reversal of the sulfate respiration pathway. Whether the attestation of sulfur metabolism and the rapid expansion of the Paleoarchean biosphere in the rock record was due to the origin of photosynthesis or the fertilization of the oceans by phosphorus is hard to say. The photooxidation of iron and its use as an electron acceptor in respiration probably displaced the earlier sulfur metabolism in the Mesoarchean and preceded dissimilatory oxygen metabolism in the Neoarchean. Overall, we can see dissimilatory redox metabolism climbing the redox ladder over the course of the Archean eon.